All right, are there any questions? So last time we were actually discussing that paper on finding out the coefficient of determination, right? And I covered a number of facts on this handout, right? The first one was that the uh, the the best predictor, if you're using the mean square error as your optimization criterion, then the best predictor in the absence of any additional data is the mean of a random variable, right? And then the second thing that I proved was that the best predictor in the presence of information or data is the conditional mean, right? That's what I finished proving right here, right? Now fact number 3. So again, I mean the conditional mean is the best predictor among all possible predictors, okay, all estimators, and this you will learn even in other classes, right? Now in general, the conditional mean is hard to compute because you need to have the conditional density function, right? Now if you are going to restrict yourself to the class of all linear predictors, right, then you can get a closed form expression, right? And that's what the next fact number 3 tells you, that if you have a random variable y, right, and you're going to make a prediction for that or an estimate of that, right, using observed random variables x1 through xn, right, then the estimate y hat, it'll be a linear combination of the random variables that you have observed, right, plus some constant here, some offset, right, and the expression for that optimal set of values that will be incorporated in a hat is given by r plus, which is the pseudo inverse of r times c, where r is the autocorrelation, right, and c is the cross correlation, right. That's the formula that you get, and this can be derived in a pretty simple fashion, because what you're going to do is you're going to figure out the error, right, and the mean square error, and you're going to try to minimize that. Okay, you have imposed the linear structure, so y will be the y hat will be of this form. It will be some linear combination of the random variables, right? So the proof is quite straightforward. Since y hat is a linear function, right? Y hat is going to be a linear combination of those random variables, right? Observed random variables which you have put in that vector v, right? And then you take the difference between y and y hat, right? And square that, right? Take the expectation. That's your mean square error, right? And then you write down the explicit expression here, right? And now you want to minimize this with respect to that vector A, right? So you differentiate this, and by now you guys know how to differentiate when you have vectors and matrices. So you get this relationship, right? And then from here you solve, you get A hat is the pseudo inverse of R multiplied by C, right? Where R is the autocorrelation, right? And C is the cross correlation, right? So this gives you the minimum mean square error. So in the linear case, you have a closed form expression, right? Now, another thing to note is that when you're trying to get the best linear estimator, right, the error that you make is going to be orthogonal to the observation, right? And that is called the orthogonality principle, right? That's proved here, right? See, and the steps are given. How you go about doing that proof. All right, but this is the error that you made, right, when you're using that linear estimator, right? That is orthogonal to the observation, because V contains all those observations. You observed x1, x2, and so on, right? This is called the orthogonality principle, right? And this is used in the derivation of a lot of optimal estimators, filters, and so on. Like if you're using deriving the Wiener filter, you'll make use of the orthogonality principle, right? And this is not surprising. Let me just show you, all right? Because if you're looking in an inner product space, all right, always, all right, the projection theorem holds, all right, in a complete inner product space. And let me just try and explain that to you, right, why this orthogonality principle is going to hold. Okay, let me just open another window and just show you why the orthogonality principle will hold. So suppose you have a subspace here, right? And you have let's say this is a zero vector. This is some line, okay? You want to project it onto that subspace, right? What is the projection going to be? It's going to be, you'll drop the perpendicular, right? So the projection of this particular vector onto this subspace is basically, so if this is x, all right, this would be the x hat, the projection, right, onto that space, all right. So what is the difference between the two of them? It's basically this line here, right? And that is orthogonal to the observation, right? See, here you're trying to estimate, let's call this y, okay? You're trying to estimate y based on measurements in that subspace, okay? And you can see that the error that you get when you take a linear combination of the vectors in that subspace, that is going to be orthogonal to the observation, right? So this holds in general in any Hilbert space, in any inner product space, and that's why we know, the closest distance, like from a straight line onto a plane, is going to be the perpendicular, right? So the ideas from three-dimensional Euclidean geometry, they generalize into these more abstract spaces, as long as it's an inner product space, as long as it's actually a complete inner product space. Okay, there is a theorem that is called the projection theorem, and this is actually a special case of that. Because when you're working with the mean square error, remember you're taking the square, right? So there's something like an inner product in there, right? So this thing holds. So this is a more general result. Again, just for your information. And if you take some of our courses on estimation theory and all that, they will cover those things in detail. Okay. Any questions on this? So now, let's get back to that explanation thing, right? So the orthogonality principle shows that the error is orthogonal to the observation, right? Next, I want to go ahead and derive a particular equation in that paper, right? So let me just pull that paper up, that COD paper, right? So we want to derive equation number 5, right? This equation here, right, in this paper. Because see, in most cases, right, you're not going to be able to calculate the conditional expectation, right? So you're not going to be able to get the error of the best predictor, right? In the unconstrained case, right? But in some special cases, you might be able to actually calculate the coefficient of determination. Remember that epsilon dot minus epsilon opt over epsilon dot, right? In order to compute that, you need to find out epsilon dot, you need to find out epsilon opt, and you need to find out epsilon dot. So this is just a special case that he discusses in the paper where you can actually get a closed form expression, right? So I'm going to show you how this is derived, right? And also how you can show that if you have jointly normal or jointly Gaussian random variables, then you can get, then basically what you end up getting is that the COD is actually the square of the correlation coefficient, right? So I'm going to show that to you. So it makes some intuitive sense. It's a generalization of the correlation idea. Correlation is between only two variables, right? And it basically is based on linearity, right? Whereas the COD, it doesn't require linearity, and it's a multivariate generalization of that, right? So let me go ahead and finish that derivation, right? So now we are going to try to derive that equation 5 in that paper, right? So suppose here we are trying to determine the best linear mean square error estimate of y given all these random variables, right? Then this A, right, that vector that I put in the previous statement of the previous fact will look something like this, right? And y hat is going to look like this, right? And without any loss of generality, because I have this constant b hat here that I'm trying to estimate, I'm going to assume that all these xi's have got zero means. Because if the means are different from zero, I can always absorb it in this one. Okay? And then here, from the previous derivation, right, you can calculate the error of the best predictor, right? That's given by this expression, and you plug in all these quantities here, you come up with this expression. Okay. Then, so here I'm just filling in the steps, right? And then you do the computation, and all the steps are spelled out here, and you find out that the COD corresponding to that linear predictor is given by this expression here. You further simplify, and all the steps are put in here. I don't want to spend more time just going through these individual steps, right? They're spelled here. If you have any questions, you can ask me later, right? I don't think you will, right? Because all of you have some basic background in doing these kind of calculations, right? This is just algebra, but at the end of the day, you will get this expression for the COD, which is the expression that is given in equation 5, right? So thus for linear prediction, a closed form analytical expression for the COD can be obtained. I am filling in the gaps here so that when you try to read the paper, you don't get lost, because over there is just a one sentence. Okay, that is telling you that this expression can be derived, right? So I'm filling in the gaps for you, right? So that's one thing. Any questions on this? Go through these steps. If you have any questions, let me know later, right? But I don't think you will, because I have explained everything in detail. Right? It's just that in the class I don't have the time to go through each and every step and crank out the algebra. Right? But all the steps are there. Now one more thing that I want to cover regarding the COD paper is that if you have jointly normal or Gaussian random variables, all right? See, if you have random variables that are jointly Gaussian, right, and you're trying to estimate one of those random variables, all right, based on the observations of the others, all right, then it turns out that the best minimum mean square estimate is in fact linear. The conditional mean actually is linear, right? The conditional mean is the best minimum mean square error estimate, right? It turns out that in that case, if you restrict yourself to the class of linear estimators, all right, you will still get the overall optimum. Okay, so and how do you show that? Well, your starting point is the joint density function. For two random variables x and y that are jointly normal with correlation coefficient rho, the joint density function is given by this, right? That you guys already know. Okay, so in this case we can find out the marginal the density functions for the marginal random variables x and y just by integrating these either with respect to y or with respect to x, right? Integrate out and you get the probability density function for x is like comes out like this, right? Which is basically the usual normal distribution with mean mu x and standard deviation sigma x, or variance sigma x squared. Now to get the conditional density function, you'll divide one by the other, right? So if you calculate the conditional density of y given x, you'll divide this by the expression that you have obtained, and you do the algebra and you get this expression. Now look, this is also the probability density function for a Gaussian, right? With a variance that is sigma y squared times 1 minus rho square, right? And a mean that is given by this thing that is sitting after the minus sign here, right? Because that's the standard formula for the probability density function of a Gaussian, right? So this is the conditional density function. So you have the conditional variance here, right? And you also have the expression for the conditional mean, right? So the conditional mean of y given x is none other than this expression here, right? The thing that takes up the place of the mean, right? So based on this, right, you can say that the condition density is a univariate normal one with mean given by that expression over there, right? And your variance given by this, right? So look, what has happened, okay? The conditional mean of y given x is actually a linear function of x because the x is appearing here. You see, it's appearing linearly, right? So in this case, the conditional mean is linear, right? And you actually have the best estimate even if you restrict yourself to the class of all linear estimators, right? And this variance is equal to sigma square y 1 minus rho square. Well, this is going to be your epsilon op. This is the error of the best estimator, right, that you can get, right? So in this case, you can actually compute the COD in a closed form. So for two jointly normal random variables, the conditional mean is a linear function of x. That is, the optimal mean square estimator is linear. And furthermore, its variance is given by this expression, which means that epsilon opt, which is the error of the estimator, which is the variance, is given by this quantity here, right? Now, what is the error of the best estimator, all right, in the absence of any information? If you don't make any observations of x, okay, what is the error of the best estimator? First of all, what is the best estimate of y, all right, without making any observations of x? Yeah, so it's the mean, right? So which means the error of that best estimate is the variance, right? So you have that expression here. So epsilon dot, which is the error in the absence of any observation, is sigma square y, and then you do the calculation, epsilon dot minus epsilon opt over epsilon dot, and you get rho square, right? So in this case, if you calculate the coefficient of determination, you see that this is nothing but the square of the correlation, right, between the two, the correlation coefficient between the two random variables, right? So the COD actually generalizes the notion of correlation to the multivariate scenario, right? And it does not restrict itself to a linear situation, right? So any questions on that? Okay, then we will close this file and go back to the paper, right? So now I have shown you how to derive equation number 5, right? See, this is equation number 5, and I also showed you the proof of this, right? That the COD is going to be the square of the correlation coefficient between y and x, right? Now, any questions, right? So I have filled in all the gaps that were needed. So here, what are we going to do? The problem is that we don't have those density functions, okay? So which means from the data, from those observations that we get, right, we are going to have to estimate the COD, right? And then this is exactly like classification, right? You first give it some samples, then you come up with a classifier, a predictor in this case, right? And then you want to also estimate the error of the predictor so that you can use those errors to compute the COD, okay? And what is the form of the predictor that you're going to assume here? We are not going to take a linear predictor. We are going to use what is called a perceptron, where, as I mentioned last time, you take a linear combination of all those variables, right? Then pass it through some nonlinear function, right? Get an output, right? And in this way, you basically make it a constrained prediction problem where you don't have too many parameters, okay? It'll be just the weights associated with the individual predictors, right? And then you try to learn that, right? So now, any questions about the methodology here? Of course, you have to read the paper to understand 100%. So the method that they developed, they actually applied to stress response data, right? And that's given in this table here, right? So when you subject cells to stress, right, different kinds of stress, like you could have ionizing radiation, you could have something that's called methane methanosulfate, right, that is toxic for the cell. You could have ultraviolet radiation. So when you subject the cell to different stresses, right, then the gene expressions will change, right? So here you have quantized gene expressions, and again using the ratio method, 1, 0, -1. So this is the data that you're going to work with, right? So across these different conditions, and there were, I think, 10 genes over here that were selected. The last two are fictitious genes, right? They call them AHA, OHO, because you wanted to see if this method is going to pick it up or not, right? So you create some rules that if this gene is high, gene A is high, gene B is low, then gene AHA will do something, right? And so on, right? So you create some fictitious rules, and then based on the data that is presented here, right? The question is, can you infer the relationships between these genes, right? So each of these genes you'll pick up, and you'll try to find out predictors. Now the question is, how many predictors are you going to use, right, for each gene? Maybe you're going to use 2 or 3, right? And remember, there are 12 genes here. If you find out predictors for them, let's say 2 gene predictors, there will be 12. Choose 2 possibilities, right? In each of those cases, you're going to go through the steps, do the perceptron and all that stuff, right? And then come up with the expressions for the errors, right? And then from that error expression, you will compute the COD, right? And see what the COD is, right? That's essentially what they have done, right? In this paper. So. And all the things that I talked to you about earlier in the context of classification, right? Like, sample size, constraint prediction, constraint classification, then, error estimation using the resubstitution estimate, cross-validation, and so on. They're exactly same kind of discussion here, right? And at the end, once you've done your analysis, you'll probably have a target gene, right, that you're trying to predict using these different predictors. If you use one predictor at a time, you get one value, right? If you use 2 predictors together, you get some value, right? And that leads us to that figure that we saw in the PowerPoint slides. Okay, that's this figure, right? I think this is P21, MDM2, and then P53. So we were saying that the COD for P21 predicting P53 is 0.227, MDM2 predicting P53 was 0.259, and the other one, when you use 2 of them together, it's 0.452, right? This is stuff that we have discussed. So now I just filled in the details to show you how they come up with these numbers, all right? So using this method, they were able to figure out some relationships that biologists already know, and something more than that, right? About the relationships between genes. But again, I should emphasize this does not give you causality, right? This is just telling you that there's some kind of a correlation or generalization of correlation between these genes. They're going up, down together. Looking at one, you can say something about the other. It doesn't tell you which gene is affecting which one. Okay. And then in the rest of the stuff in this paper is, I think they developed some software, some user-friendly software that will allow you to, run this kind of COD analysis, all right? And then it'll spit out which are the genes possible predictors, and you can do all kinds of things, So it's just showing items on the that can be displayed. And I don't even know whether that software is being used right now or not, okay? Because this paper is a 2000 paper, right? So, but this information, this COD information of association between genes, right, it can be used to find out the predictive relationships between genes, right? Because here, this is different from many engineering systems where, the cause and effect is kind of known, right? Here you don't know. It's just, you're just having those snapshots, and based on that, you're trying to find out relationships. And then the last section of this paper is about statistical considerations. It's more mathematical stuff, right? If you're interested, you can take a look. But I just wanted to point out one thing over there, that the update rule for the perceptron, right? It's like neural network training, right? This appendix discusses this update rule for the perceptrons. And I bring it up because somebody asked me about this in the last class, how do you update the weights of the perceptron, right? It'll be some kind of a gradient descent algorithm, right? And this is the update, this is the error, right? And then there is an update formula, right? How you update the weights of the perceptron, right? But again, I mean, this is standard stuff, this is well known, but somebody asked me last time about that, but he's not here in the class today, so I just wanted to point that out, right? Okay, any questions? But again, I mean, to understand everything in this paper, right, you have to read it. You have to spend some amount of time. And I'm not forcing you to read it. If you're interested in doing research in this area, you'll go and probably spend the winter break reading all these papers, that we are discussing. And I'm just highlighting the key features, trying to explain the big picture. The nitty-gritty details you can go and read. Right? I don't have time to cover that here. Okay, any questions? So using the methods in this paper, you'll basically be able to figure out, given a particular target gene, right? You'll be able to figure out which are the best predictors for the genes, right? Let's say which two other genes predict the activity of this gene pretty well, right? In other words, looking at those two genes, can I say anything more about this gene that I'm interested in, this target gene, more than something that I already knew, right? Because the best predictor in the absence of any information is the mean, right? The best predictor when you have that information is the conditional mean. The problem is we cannot calculate those, right? And therefore we have to learn these things from sample data, right? That's what's being done here. So any questions? So now we will go back to our PowerPoint notes and proceed from there. All right. All right, so if you're given a target gene, right, using that COD method, using the method of that paper, right, you may come up with several predictor sets, right, that predict the activity of that gene quite well, right. So if I had only one set of predictors, then I would get a Boolean network because I would try to get a Boolean function. Remember, in the Boolean network, the activity of the ith gene at time k+1, right, is basically a Boolean function of its predictor set, right. So now it might turn out that there are lots of good predictor sets for a single target gene, right? And I don't want to put all my faith in one predictor set, right? So then I can say that, okay, the activity will update according to the first predictor set with a certain probability, right? Second predictor set with a certain probability and so on, right? And it would make intuitive sense to do the update in the ratio of the CODs, right? Because the COD is measuring the goodness of a given predictor set. It is telling you how good that predictor set, right, is for predicting that particular target, right? So that's the key idea of the probabilistic Boolean network, right? And I'm going to talk a little bit about this later on, right? So I'm going to kind of go a little bit faster over this stuff. Let me see. So yeah, as I said here, while attempting to infer intergene relationships, it makes sense to not put all our faith in one predictor set, Instead, for a particular target gene, a better approach is to consider a number of predictor sets with high CODs, all right? And considering each retained predictor set to be indicative of the transcriptional activity of the target gene with a probability proportional to its COD, right? That makes sense, right? I mean, because if the COD is high, that means that's a better predictor, right? So I'm going to put more weight on that. So having inferred these intergene relationships in some manner, this information can be used to model the evolution of the gene activity profile over time. And so by associating several predictor sets with each target gene, it is not possible to obtain with certainty the transcriptional status of the target gene at the next time point. However, one can compute the probability that the target gene is going to be transcriptionally active at time t plus 1 based on the gene activity profile at time t. So now it's not deterministic, it's not a deterministic Boolean network anymore, right? It's a stochastic dynamical system, right? Or it's a generalization of a Boolean network, right? With stochasticity introduced, and it's called a probabilistic Boolean network, right? So the time evolution now defines a stochastic dynamical system. And since the gene activity profile at a particular time point depends only on the profile at the immediately preceding time point, right? The dynamical system is Markovian, right? Because given the current information, you can forget all about the past, right? As you're trying to predict the future, right? So such systems can be studied in the established framework of Markov chains and Markov decision processes. And for those of you that have taken Professor Kumar's course, he spends like the whole semester talking about these, control of Markov decision processes. All right. And if you haven't taken, that's, as I said last time, that's a good course to consider next semester, right? So these ideas are mathematically formalized in probabilistic Boolean networks. So in a probabilistic Boolean network or a PBN, the transcriptional activity of each gene at a given time point is a Boolean function of the transcriptional activity of the elements of its predictor sets at the previous time point, right? So the only difference is that here for one target gene, you could have several sets of predictors, right, with different probabilities. And the choice of the Boolean function and the associated predictor set can vary randomly from one point to another, right? In which case the network is changing at every time point, right? On the other hand, you could have a situation where the network stays the same for a certain amount of time and suddenly it switches to something else, right? It becomes a different network, right? So there is a distinction between what is called an instantaneously random PBN, right, and a context-sensitive PBN. In fact, I will talk more about this in a later lecture, so let's not worry about this here, right, because there are pictures and things that are coming. Probably it'll be next time, right, when we talk about that, right? Yeah, this is about the context-sensitive probabilistic Boolean network, right? Uh, let's see. Now this business of the probabilistic Boolean networks, it can also be generalized to the case where you have ternary data, right? Because normal Boolean logic is 0, 1, right? I mean, but you could actually use 0, 1 and minus 1, right? So, I mean, there's nothing that stops you from ternarizing that logic, right? So this could be generalized, right? And yeah, let's skip all of this stuff because there are some examples that I'm going to show that is going to clarify all of this stuff here, right? Uh, yeah, let's not worry about this, right? This I already mentioned about the Markov chain, right? Then just like in the case of a Boolean network, right, we had the attractors that were characterizing the long run behavior, right? In the steady state, you end up in an attractor, and I showed you a picture last time of that attractor, and there were a number of levels that you go through to get to that attractor. Here the only difference is that things are stochastic. So instead of attractors, what are you going to have? Anybody took Professor Kumar's class or nobody? All right. You will basically have you'll basically break the state space up into different equivalence classes where different states communicate, right? If you didn't take his class, don't worry about it, okay? In the example, when I do that example, I will point out, right? Because this is all general theory, but the important thing to note is once you have put it in this framework, right? The theory is already there, out there in the literature, in the controls operations research literature. That theory is there for controlling Markov chains, all right? The problem here is how do you go from the biology to the Markov chain? It's easy for me to put it down over here and to say that intuitively all this will work, okay? But have you validated your network, right? Before you try to control and intervene and do things over there, that network needs to be validated, right? And that is not easy to do. All right, so let's just skip this part. All right, now the other thing is that you're going to have to infer the network, right? See, once you have a probabilistic Boolean network, you need to learn that network from the data, right? So how do you learn that network, right? So one approach could be to use the COD, right? Use the COD to find out those probabilities that you're going to get associated with each of the predictor sets, right? There are other approaches also people have used, right? And each one of them is a paper, and I mean these have been published about 10 years ago. They are listed in our book, right? The references at the back, you know, those papers are listed. If you're interested, you can look at them. But again, I mean, as I said, this is like the stuff that is like almost 10 years old, right? But the important thing to note is that here you're looking at an ill-posed inverse problem because you are going to collect data in the steady state. You do not have time course data. This is different from trying to identify a system, right? Where you're looking at its behavior over time and then you're trying to fit a model. Here in the biological case, if you run a microarray, it is with steady state data, right? So there might be lots of models, lots of networks that might fit that steady state behavior, right? So this is an ill-posed inverse problem, right? You're trying to identify the model, but the problem is that the solution is not unique, right? So what you might have to do is you might have to put in constraints, right? If there are some biological constraints that you know, right, about how the genes behave, you will have to kind of use them to constrain the solution, right? So that's what is going to basically make your network more biologically relevant, right? And it is going to enforce some structure, and it is also going to reduce the class of networks, right? So I will talk about this, as I said, you know, when I do the examples, right? When I'm going into intervention and control, I will talk about those aspects, you know, at least try to give you the big picture. And again, if there are any questions, if there are any gaps in whatever I'm saying right now, feel free to interrupt me, right? It's just that I don't want to, you know, go through all of these details here because it is going to take a lot more time, and you know the notes are themselves were probably prepared like 10 years ago. So there are some new things that I want to bring in, right? Because I know several of you here are in the research mode, right? You want to quickly get into research. It's good to look at those things also, but I want to show you the latest developments and what the latest thinking is, so that you can take off from there, right? And I'll do that using some examples. Yeah, so this is what I talked about, the PVN inference using the COD, right? So, so the selection probability of each predictor set for a target gene is assumed to be the ratio of the COD of that predictor set to the sum of CODs of all predictor sets used for that target gene, right? It makes sense because the COD is measuring the goodness of the predictor. So you basically will make the transition according to that predictor, right? With probability proportional to the COD. Right? So that was the first one, and then there are other methods that have been discussed. For example, PBN construction using mutual information clustering and Markov chain Monte Carlo technique, right? And again, you may have come across. How many of you here know what mutual information is? You know, okay. So the communication guys know. So you guys, the rest of you, if you want to learn about mutual information entropy and so on, right? Then you might consider taking some of those communication classes. See, that's why I told you guys that, you know, your communication stuff is not going to go waste, right? It is related, right? But you have to learn the biology first before you're going to do the work here. So take those courses also, right? Because you don't want me to be teaching you these things. I'm not an expert in all this stuff. The mutual information, again, it's something similar to this COD thing, right? It's telling you about the coupling between variables. Okay, how much of information can I get about this variable looking at that one, right? But again, it's made mathematically very precise, and so you need to take those classes, right? Okay, yeah. So one more thing that I want to talk about is, you know, instead of building these networks just looking at the data, maybe you have some prior knowledge, okay, that there is a particular gene, let's say P53 is very important for something, all right? So you want to build the network around P53, right? So you might know that for some particular biological phenomenon, all right, there are like 3 or 4 genes which are very important, right? That they play a central role. So you might want to seed your algorithm with those genes and then start growing your network, right? And here is an example of that, right? So this is in the context of melanoma. These genes are important: WNT5A, SNCA, S100B, RTN1. These are very important genes in the context of melanoma. The biologist has given the authors of that particular paper this information that these 4 genes are important. So now you're trying to grow this network, all right? Using the data, you're trying to grow this network starting with these seed genes, right? And there is a paper out there that was published many years ago, maybe 2004 or 5, right? I was not involved, how to grow this network, right? And once you grow this network, again, I mean, it's not definitive, because this network does tell you some information that biologists knew from before, right? And they're able to explain it, but then there are some other places where there are connections, right? But they didn't know from before. But at the same time, they cannot rule out that there are no connections, because this stuff is really quite complicated, right? It's just because somebody else has not seen a relationship, right, does not mean that that relationship does not exist, right? So in that sense, this method is not going to be like that definitive, but this is yet another approach for trying to come up with a genetic regulatory network, right? Starting with some seed genes. So that's the melanoma network, starting from those 4 seed genes. And the criterion they use for adding these other guys is using COD. If the COD is more than a certain threshold, then they added these other nodes here, right? Like this one is very important. MAP2K is that's the mitogen activated protein kinase pathway, and I will talk about that later on, right? It's involved in the control of cell division. Remember, we had all those kinases, right? Cyclin dependent kinases and all that stuff, and those are controlled by the mitogen activated protein kinase pathway. Yeah, here it basically says that in some cases, once they got this big network grown from the seed, they were able to relate it to some things they knew, and then there were other cases where they were not able to relate it, but it's possible that some kind of relationship exists. Right? It's just that we don't know as yet. Yeah, this I already mentioned, the issue about network inference from the steady state data. Right? You do not have time course data. Right? For biologists, even the time course data, it might be like you have a population of, let's say, mice, all right, and you kill some of the mice at different time points, all right. So it's coming from different animals. That is their time course data. It is not like what we do in the lab where you have a control system or something like that. You're trying to identify the same system you're running over time. You're getting time. They don't have that kind of time course data, right? So you have to work with steady state data, which again makes the inverse problem ill-posed. Right? So again, I mean, different approaches that were taken, and most of these are from our group, all right? People published these papers, all right? And again, it's just some method that is already used in engineering or statistics, all right? You applied it to genomic data, right? And there is extensive discussion of all this. And then the last thing I want to talk about in this particular chapter is what are called Bayesian networks, all right? So Bayesian network basically it gives you the conditional probabilistic relationship between random variables, right? And this has been extensively studied. So there is a version of that Bayesian network which is called a dynamic Bayesian network, right? And that is also studied quite a bit, right? Now, what they have shown is that the probabilistic Boolean network, right? There's a relationship between the probabilistic Boolean network and the dynamic Bayesian network. And again, that's another paper that somebody has done, right? So, see, there is a precisely characterized relationship between certain dynamic Bayesian networks and probabilistic Boolean networks, in the sense that they can represent the same joint distribution over their corresponding variables, right? And PBNs are more specific in the sense that the mapping between PBNs and DBNs is many to one. So that if you have a dynamic Bayesian network, it is not going to specify a unique PBN, right? So PBNs can be analyzed using Markov chain theory, right? Which means that even dynamic Bayesian networks, you could convert them into a PBN, right? You can go from one to the other and then use the power of the analysis machinery for Markov chains to carry out the analysis, right? So somebody has shown this equivalence between the two of them. Starting from a PBN, you go to a DBN and vice versa, but it's not a one-to-one map. Okay. All right, any questions? Any questions on this design of regulatory network? So again, the COD is important, right? And then there are a lot of other papers. So if you look at I have given you a list of papers in the syllabus, right? Like 1, 2, 3, I think it was up to 8 maybe. There were 8 papers listed. The first 2 papers I already discussed here, right? The third paper is about probabilistic Boolean networks, all right? Where, you are actually selecting those predictors, right? according to their CODs, right? That's the third paper, right? The fourth and fifth paper are the intervention that I'm going to discuss right now, where you have a network, all right, and based on that you're trying to, intervene. The network is not behaving in a nice way, right? Like it's going to a bad attractor. How do you try to induce the network to go to a good state, right? That's the question that we will look at, right, in this particular chapter. So any questions on that? So this chapter we will basically see how you can intervene in the network, right? Either by moving the state out from an undesirable location, right? The state has ended up in a bad situation. Let's reset it to some other location, and then hopefully things will get taken care of. Or change the wiring diagram of the network, right? Those are the two things we'll look at here, right? And in the next lecture, that is next Tuesday, I will look at how you can use the theory of Markov chains to actually control these probabilistic Boolean networks, right? And also I will show you how to incorporate prior information, right, into the design of the intervention, right? So I'll show you all that stuff next time. So today I want to get through this chapter, and I will get through this chapter using an example, right? Because the example will make everything crystal clear, and that example is borrowed from those papers that are listed in your syllabus, right? So if you want to know more about this stuff, then you have to go and read those papers, right? I mean, each paper will probably take a week to discuss, right? Week or at least one lecture, so I don't have time to discuss all of them, right? But again, I mean, you're engineers here, so for you the difficult part or the stumbling block is basically the biology, right? And my goal is to focus on that more, right, in this class. Both animal biology, basic molecular biology, organic chemistry, cell biology, and then plant biology, right? This stuff is kind of easy for you. The papers are there. I'm going to give you the big picture, right? So intervention, the ultimate objective of genetic regulatory network modeling is to use the network to design different approaches for affecting network dynamics in such a way as to avoid undesirable phenotypes, for example, cancer, right? So the pragmatic manifestation of this goal is the development of therapies based on the disruption or mitigation of aberrant gene function contributing to the pathology of a disease, right? So mitigation would be accomplished by the use of drugs to act on the gene products, right? So engineering these therapeutic tools or designing them involves synthesizing nonlinear dynamical networks, analyzing these networks to characterize gene regulation, right? And developing intervention strategies to modify their dynamical behavior, right? All that makes sense, right? I mean, that's what we do in control engineering all the time, right? We're trying to model the system and then come up with a controller that will hopefully make the system behave in a desirable way, right? So this is nothing different here, except that now the focus is on treating the phenotype, right? If the cell is producing tumor, too much of signaling, we want to cut down on that, right? So if you have a network that is functioning normally, right, there could be mutations or something in that network because of genetic mutations that is introducing additional connectivity or something like that. So now the steady state the steady state attractors of that network now involve continuous signaling to promote a tumor or something like that, right? So the intervention studies using probabilistic Boolean networks have used three different approaches, right? The first thing that you can do, and that's the easiest thing, right, that is reset the state of the probabilistic Boolean network to a more desirable initial state and let the network evolve from there. So here you don't try to re-engineer the network. Let the network remain. Maybe for some reason the state has ended up in a bad location, right? I just force it back to a good location and I hope everything will be fine. Right? It may not always be, but that's one approach. Another approach might be to change the steady state behavior of the network by minimally altering its rule-based structure. Remember, when you're getting this network, this probabilistic Boolean network, you have a bunch of predictors. You have a truth tables, okay, that are telling you that if you are in this state, these are the states that you will go to with these probabilities, right? So if I go and alter that truth table, right, I might be able to alter the distribution of the good and bad states, right? I might be able to put more probability mass into the good states, right, and less probability mass into the bad states, right? So these are the two things that I'll cover in this chapter today, right? And then the last one is manipulating external control variables that will alter the transitional probabilities of the network. That is the probability of the network going from one state to the other, right? That will depend on external variables. Like if you're treating cancer, you're giving chemotherapy, you're giving radiation and so on, right? The evolution of the genes is not going to remain the same, right? When you apply treatment versus when you don't do anything, right? So the treatment has a cost associated with it because when you try to treat a cancer, If you overtreat, you'll probably kill the patient, right? So you have to be careful, and even the drugs are also expensive and so on, right? So this would be an optimization problem, right? To treat or not to treat, or how much to treat, right? So I will talk about that next time. This item number 3, but 1 and 2 we can discuss today. Before that, any questions? So in this lecture, we discussed the intervention results obtained using the first 2 approaches. The results obtained using the third approach will be discussed in the next lecture, right? So given a probabilistic Boolean network, the transition from one state to the next takes place in accordance with certain transition probabilities and their dynamics, right? Because you are saying that, okay, gene number 1, right, is going to transition to certain state. If it is in a given state, it will transition to some other state with a certain probability, right? So based on that, you can compute the transition probability of the associated Markov chain. And so intervention can be studied in the context of homogeneous Markov chains with finite state spaces, right? Let's say if you have n genes, right? You're looking at n genes. Each gene can be either on or off. So how many states are there? How many possible states are there? 2 to the power of n, right? Possible states, all right? And so now, so it's a finite state space, all right? And so you can look at this as a control problem, right? Where you're trying to move the genetic regulatory network, right, from a bad location to a good location, right? And you have to actually properly do that classification. So now I want to show you how you can go from a probabilistic Boolean network to a Markov chain, right? Remember I mentioned in the last chapter that I'm going to show that to you, right? So to characterize the Markov chain associated with an instantaneously random probabilistic Boolean network, let's first focus on Boolean networks. Let's forget about all the stochasticity right now, all right? Because if I cannot do something similar in the case where there is no randomness, there's little or no hope that I'll be able to do any better when I inject some randomness into the picture, right? Now for Boolean networks, the state vector xk at any time k is essentially an n-digit binary number. If you have n genes, it's an n-digit binary number, right? Ones and zeros in all the locations, ones or zeros. And you can compute the decimal equivalent by this formula, right? I mean, given a binary string, you can go and compute its decimal equivalent. So as xk ranges from 0 all the way up to n ones, all right? Yk was going to take on all values from 0 to 2 to the power of n minus 1, right? And let's define a new variable zk to be 1 plus this yk that you have calculated. That means that xk ranges from over all these possible gene activity profiles, all right, or patterns, all right? Zk will take on values from 1 to 2 to the power of n, right? So there are 2 to the power of n states. Now the mapping from xk to zk, right, from this binary to its decimal equivalent plus 1 is 1 to 1 and on to, and hence it is invertible. So you haven't lost any information. So thus, instead of the binary representation, xk for the state vector, we can equivalently work with the decimal representation zk, for which the state space now becomes 1, 2, 3 up to 2 to the power of n, right? Now each of these zk's, right, you can represent them uniquely by a basis vector, because you have 2 to the power of n vectors, right? Okay, the first vector, when you have like all zeros, okay, which is mapping into 1, I will just represent it as 1 and then all zeros, right? Then the next one I'll represent as 0, 1, all zeros. Next one 0, 0, 1, all zeros, right? So again, now I have 2 to the power of n vectors, right? Right? And each of those is 2 to the power of n dimensional. So the evolution of the vector, now with time the vector is going to change, right? I mean, as the state evolves. So the evolution of the vector will proceed according to this kind of a difference equation. Because you have wk, that's where you started out, right? If it's 1, all zeros, that means you started out at the gene activity profile, all zeros, right? Where will you go from there, right? If it's a Boolean network, that's a unique place that you will go to, right? So the elements of this matrix A will be chosen to decide, to basically make sure that you go to the correct vector, right? So this A is going to be a 2 to the power of n by 2 to the power of n matrix, right? Because the state vector is 2 to the power of n dimensional, all right? And it is also going to be having only one non-zero entry equal to 1 in each row. So each row you will have just a 1 at some location and zeros everywhere else. Alright, so that's how you're going to map the state at time k to the state at time k plus 1. Right? And again, you can check this out and verify that what I'm saying here is correct. Right? But this is deterministic because if you start out from WK, so WK will be everywhere it's 0 except for a 1 in one location, right? So all the probability mass is over there, right? And then from there you'll go to some other state. With probability 1 you'll go to some other state for sure, right? So again you'll have a 1 in a particular location, zeros everywhere else, right? And the location where you have that 1 will be determined by the entries of this A matrix, right? So now I want to generalize this to the case of probabilistic Boolean networks. Well, probabilistic Boolean networks, what is going to happen? If you are in a particular state, you will transition to a number of states with different probabilities, right? So the difference is instead of this WK being just a vector with 1 and all zeros, you will have a number of different entries, positive non-zero entries, right? Sorry, non-negative entries that will have to add up to 1, right? Because that is going to be the probability distribution vector, right? Or what Professor Kumar refers to as the occupation probability, right? So that's what that vector is going to be, right? And so this is going to be the occupation probability vector at time k, occupation probability vector at time k plus 1, right? And then this matrix is going to be what is called a stochastic matrix of transition probabilities, right? With every row summing up to 1, right? So this equation that we derive for a deterministic Boolean network has a stochastic counterpart for a PBN. To arrive at the counterpart, let Wk denote the probability distribution vector or the occupation probability vector for a PBN at time k. That is Wik is the probability that at time k z is equal to I, right? Then it can be shown that Wk evolves according to this equation. Wk plus 1 equals Wk times the matrix A, where A is the stochastic matrix of transition probabilities. So the stochastic matrix will have the property that all the rows, all right, are non-negative, all right, and each entry is less than 1 and everything sums up to 1, right? And so we are now able to go, right, from a probabilistic Boolean network, we are now able to go to a Markov chain. So all the theory that is available in the literature for the control of Markov chains, right, all that becomes immediately applicable, right? And here all this tells you is that just like we've done binary quantization, the same kind of stuff could be done in the ternary scenario too. But we'll not worry about that right now, right? Now, so our goal here is to basically intervene, right? As I said, we have a network, we may not like where we are in, right? We want to intervene and make the network go to a good state, right? And I think this will be best illustrated using an example, okay? So suppose, right? So this is the example, and this is taken from the, let's see, from the third paper, I think. No, this is taken from the fourth paper in that list, right? The third paper is basically just introducing probabilistic Boolean networks. Fourth paper is discussing intervention in Boolean networks, the fourth and the fifth one. So suppose you have a PBN consisting of 3 genes. We want a simple example, right? 3 genes that can take on binary values. So how many states are you going to have? No, there are 3 genes, so 2 to the power of 3, 8 states, right? So you will have 8 states and then there will be a truth table, right? So see, x1, x2, x3, 3 genes, you will have all zeros, all ones. So there is 8 possibilities here, right? This example is taken from that paper. And then you have predictors. So you will have, for each gene, you're going to have a number of predictor sets. So for gene number 1, you have 2 predictor sets. So this is the truth table for the first predictor. This is the truth table for the second predictor. This one is selected with a probability of 0.6. All right, let's say you did COD analysis or something like that. This one is selected with a probability of 0.4. So the two have to sum up to 1, right? Because these are the two possible predictors for the same target gene. For the second gene, you have only one predictor that's given by this, and so the selection probability is 1. And then for the third gene, you again have 2 predictors, and these are the values that you have. So if you are in state 0, 0, 0, right, you will basically transition. You look across here. You will transition with probability 1 to only 0, 0, 0 again, okay? Because for all these genes, if you are in 0, 0, 0, no matter which predictor you choose, you're still going to stay in 0, okay? So that is going to be like an attractor. Same way, if you start at 1, 1, 1, you're going to just stay in 1, right? You're not going to transition anywhere. So using this information, right? Using this information in this truth table, right? You can actually draw the transition diagram for this particular network, right? See, if you're in 0, 0, 0, you transition back to 0, 0, 0 with probability 1. If you're in 111, you transition back to 111 with probability 1, right? For these other places, you will transition to different locations with different probabilities. So C1, C2, and C3, they are coming from here, right? So I don't know which one he calls C1, but you can work out the details, right? But this is the structure that you will get, right? Now, so this is a probabilistic Boolean network because you're not definitely making this transition. You're definitely making, but these you're making with certain probabilities. If you're in 0, 1, 1, with certain probability you'll go here, certain probability you'll go here, with certain probability you'll go here, with certain probability you'll go here, right? And of course, all these numbers have to add up to 1. Okay, so this is like a Markov chain. This is a Markov chain, right? However, this is not. See, if you have a Markov chain, if all states you can go from any state to any other state, all right? Then you say that all states communicate, right? This is not a Markov chain where all states communicate because you can see that if you are, let's say, in this state, right? There's no way you are going to get here. See, because all these arrows are going in this direction. There is no arrow that is coming out from here going over there, right? So now if you have a communicating Markov chain where all states communicate, then you can show that if you run that Markov chain, there is a steady state distribution. That means in the steady state, the probability, you can, what happened? In the steady state, all right, you can compute, there'll be a fixed probability of being in any particular state, right? Because among these 8 states, you'll get a steady state distribution. You can compute that, right? If you have a Markov chain which they say says ergodic, which means all states communicate. This Markov chain is not ergodic, right? Because if you get in here, you just cannot get out. Okay? Now suppose, right, that this guy is a bad state. All these genes high, that is bad, and this is a good state, right? And you want to get there, right? So how do you get from here to here? From this chain you cannot get, okay? You have to do something. You have to tinker with one of these genes over here, all right? So let's say you have the flexibility to use a drug or something to go and flip one of these genes from 1 to 0, right? And the question is, which gene should you use, right, to do that flipping, right? So you can answer that question in a number of different ways, right? So first let's look at it intuitively, right? So let's say you are in state 111, you want to go to state 000, right? So reach state 000 starting from state 111. Why? Because for some reason, based on biological knowledge, we know that this is a bad state and this one is a good state. I want to get there, right? And this is the paper where this example is taken from, okay? I think it's the 4th paper that is listed in your syllabus. So I can try out, so there are 3 possibilities here. I can flip the first gene 011, right? Flip the second gene, I'll get 101, or flip the third gene, I will get 110, right? Which one should I use? Anybody? 011, yeah. Why? Because with the others, and let's go through the steps, all right? So if you flip 011, you will come here, so there is some probability of going back here. No guarantees, but there's some probability, right? So there's some hope, all right? On the other hand, if you flip the second gene, you got, yeah, you came to 101. From here, there's no way that you'll get out, right? I mean, because everything from here is pointing this way. There's no way to get out. And the same is true for the other gene. Flipping gene number 3 got you to 110. You're still stuck in that same equivalence class, right? You cannot get out of there, right? So based on this observation, right? Again, we didn't do any math. We just flipped things, all right? So the states 110 and 101 will always return to state 111 with probability 1 as there are no outward edges. Hence, gene 1 should be flipped, right? The question is there a more rigorous procedure to come up with the same answer, right? And the answer is yes, you can use the theory of Markov chains to do that, okay? So before doing that, right, I have to make a small assumption, because in this particular chain that I have, all right. See, if I go back to this diagram, all right, here, see, if I want to use the theory of Markov chains, if I went and manually flip that, right, one of the genes, then I came back here. Otherwise, there's no way to get out of this, all right. So I'm going to change the Markov chain a little bit. what I'm going to do is that I'm going to say that at any state, when you're in any state, there is a small probability, very small probability of a perturbation, right? That might throw you off from here and like, let's say get you here, right? So what is going to be the result? If I allow a perturbation, so any gene, right, if it's at a particular state 1, there is a small probability, very small, with a small probability it might flip from 1 to 0, right? If I make that assumption, then what happens is that you can go from, it might be with a small probability, but you can go from any state to any other state because there is that probability of perturbation, right? Because this is without any perturbation, but if I allow perturbation, there is a possibility that this state number 1 could flip here, right? It could become 0 and then from here you could go there, right? So I can make all states communicate. How? By putting in an artificial probability of perturbation. Because then it makes the problem mathematically simpler because then all states can communicate. You can go from any state to any other state and I can actually compute how long it takes me to go from one state to the other, right? So I can compute what is called the passage time, right? The first passage time and so on. And these are ideas that are well studied in the theory of Markov chains, right? So I will put in, on this network, I'll superimpose a small perturbation probability P, let's say of 0.01, right? And in that paper that is the one where PBNs are introduced, all right? And even in the next one, they have done all the calculations because when you put in that perturbation probability, okay, what do the new transition probabilities become? So the closed form expressions are calculated over there, right? What these different transition probabilities will become when you stick in the perturbation, and they can also calculate, in that case there will be a steady state distribution because all states communicate, right? So and that is also computed, right? So if you want to use the theory of Markov chains, instead of just looking at this figure and just flipping one gene and then trying to see, here we got lucky because in those other two cases the thing was stuck in there, right? So if you want to avoid this situation and get a systematic procedure, so then we introduce that random perturbation. So assume that there is independent random perturbation with little p greater than 0 so that the Markov chain is ergodic. Ergodic means all states in the Markov chain communicate. You can go from any state to any other state and every state will eventually be visited. Now the question of intervention can be posed in the sense of reaching a desired state as soon as possible. All right. So once I make that Markov chain ergodic, all right, this chain ergodic, all right. So I want to see how quickly I can go from here to this one, okay? So now the question is, if I flip the first one, 0, 1, 1, right? I'll end up here, right? If I flip the second one, I'll end up here. And from each of these, I can calculate what is the mean first passage time, right? The first passage time is, you can compute the probability of going from this to this state, right? In one time step, right? Two time steps and so on, okay? And you can calculate the mean. What is the mean first passage time, right? So let me elaborate on those ideas, all right? So that's one way to do that, right? So let's just go through this text, all right? So for instance, in example 1, if p is very small and we are in state 1, 1, 1, then it will be a long time until we reach 0, 0, 0. And setting the first gene to 0 is much more likely to get us there faster. So hence we are interested in this probability fk from x to y, that starting in state x, the first time the PBN will reach some given state y will be at time k, right? We are interested in this probability, right? So this is known as the first passage time, right? The first time you reach some other state, right? Starting from state x. If k happens to be 1, then this is nothing but the transition probability. This is telling you what is the probability of going from state x to y in one time step. Okay. So now, so you can actually examine your results by considering this following function, hk0xy. So this is basically adding the probability, all right, of going from state x to y, all right, in k0 steps or fewer, right? So if you go in the first time you hit y, right, if it is in one time step, that corresponds to k equal to 1. That's the probability, right? If you hit state y for the first time in 2 time steps, then this is going to be with k equal to 2. And notice that these are mutually exclusive events. You can hit it for the first time either after step 1, all right, or step 2, all right? You cannot hit it for the first time after step 1 and again hit it for the first time in step 2, right? It's already gone. So these are mutually exclusive events, so you can add up these probabilities, all right? So what I could do, right? So what I could do, I could try to maximize, so given a starting state X, all right, if I'm allowed to flip a particular gene, right, if I flip gene number 1, it's getting me a new starting state, right? If I flip gene number 2, another starting state. From each of those starting states, I can see, where is the probability going to be maximum, right, of hitting that destination, right, in k time steps or less, right? I can compute that, right? That's one possibility. Another possibility would be to look at the mean first passage time, because the passage time was k. So if you want to compute, right, the mean first passage time, this is the basically computing the expectation of any discrete random variable. This is the probability, this is the value, this is the mean first passage time. So that's the average time it will take to get from state X to state Y, right? So you might try to find out, when you start with the state, with that state, flip each of those genes, right? From the new state that you get, right, to the ultimate destination, which one has the smallest mean first passage time, right? That'll be the gene that you will select. So for this example, any questions so far? Again, things will become clear as I work through this example, right? So continuing with this example, it is possible to compute the entries of the matrix A. So suppose the perturbation probability is 0.01. In that case, the steady state distribution is given by this vector. Okay, so this is for state 000 and this is for state 111. You can see that the probability mass in 000 is quite small. In 1, it's quite high, right? And so the PBN is spending more time in a bad state, right? So let our starting state be 111 and the destination state be 000. The question is, should we intervene with gene 1, 2 or 3, right? So we basically compute this FK for each of these situations, right? So if you perturb the first gene, you got 011, so compute this. Then compute when you perturb the second gene 101, and then compute this when you perturb the third gene, so that's 110, right? And you compute this stuff and you calculate that HK0, right? XY, which is the probability of hitting Y in k or fewer time steps, right? So then if you compute that, if you run through all the computations and you calculate these quantities, these are the three graphs that you get. So this is the plot of HK0, right? And this is for the three situations, 011, 110, 101. So which one should you use? You should perturb the first gene, because the probability is the highest. Because if you flip the first gene from 1 to 0, then you're getting much higher probability, right, of hitting that 000 in, I think, 20 steps or fewer, right? So this is giving you the same answer, but you are using a more precise mathematical method, right? Not just trying to look at the graph and see how it's going, right? Yeah.
So does this graph says that for the 011 after the first try only we will reach there? What is the significance of k0?
No, this graph is telling you, so if you allow more time, okay, it is telling you what is the probability of reaching there. It's not telling you anything for sure. Okay, what is the probability of reaching there? Let's say in 2 time steps. Okay, that's what it is. In 4 time steps, that's what it is. 6 time steps, that's what it is. Okay, of getting to 0, 0, because you have summed up. If you are going to reach in 6 time steps, that means you could reach in 1 time step, 2 time steps, 3, 4, 5, 6. Okay, so in 20 or fewer time steps. So this is telling you the probability. If you started at 0, 1, 1, this is the probability of reaching in 20 time steps or fewer, okay? Here also is the same thing, right? And you can see that this is highest, right? Probability of...
Probability of not reaching is still high, right?
No, it's still, yeah, it's still high, yeah. Probability of not, maybe you have to wait longer, right? Yeah, that's still high, but you don't have that much of a choice. These are the three possibilities, okay? There's this and this, okay? So
Longer might not help. I mean, we might not very well reach a probability of 1.
yeah. I mean, again, I mean, you'll have to do the detailed analysis, but I'm just saying that here, this is among, it's like among all choices that are not good, you're picking the best choice, okay, right? So this is one way to do it, right? The other way is to use the mean first passage time, right? So this is basically discussing all the stuff that I mentioned, okay, that probability is almost 0.25, so you should intervene with gene X1 rather than with X2 or X3. Now, if you were to base intervention on mean first passage time, then the best gene for intervention would be the one which possesses the smallest mean first passage time, right? Agree? All right? So if you, and again, these are things you can calculate, okay, because in the theory of Markov chains, there are ways of calculating all this stuff. So because you have the transition probability. So if you calculate that with perturbations of genes x1, x2, and x3, that means x1 means you're looking at the state 0, 1, 1, right? What is the mean first passage time from 0, 1, 1 to 0, 0, 0? You do the calculation, you get this number, so many seconds, all right? For the other one, you get a bigger number, and the third one, you get an even larger number. So even if you use mean first passage times, you come up with the same result, that you should intervene with the first gene, right, to in order to make your intervention most effective, right? All right, any questions?
So how's the selection done for the mean time?
How is the what?
How do you select the time? Is that chosen probabilistically?
No, the mean first passage time you can compute, right? I mean,
Yeah. So, I mean, how does the system recognize which one is the better time?
no, this one you'll compute, right? I mean, you'll do some calculations, right? You're not running in anything, you do some calculations, right? Because you're in state A, state B, okay? So you're looking at the probability of, hitting that state in, let's say, 5 time steps, all right? Is something. In 1 time step is something. In 1 time step, it is the transition probability. For 2 time steps, you can calculate. Then 3 time steps. So it's a probability computation, right? Once you have those probabilities, you can then calculate the expectation, right? That'll give you the mean, right? Mean is the expected value, right? The expected first passage time, right? You can calculate that. That's a probability calculation, right? So you have to work that out once you know the transition probability, and based on that they got these numbers, all right, 337.51 and so on. But the point is that this is smaller than these other two, so you will go and intervene with the first one. Okay.
I mean, we are making the decision on that. That's,
Yeah. You're making the decision based on these numbers, because it's saying that the average time for reaching that, so you will reach that state faster. But again, here nothing is guaranteed, okay, because everything is stochastic, all right? It doesn't mean for sure, okay? But on the average, you're going to reach there faster, right? So that's how you're making the decision. All right, now, so any questions on this that I covered, right? So this actually is discussed in detail in one of those other papers, okay? I think it's the fourth paper, right? And the next thing that I'm going to talk about, that is the fifth paper, okay? Now, see, again, I mean, like, let me go back to that picture and say something, and then I can explain very easily, right? So remember this example, all right, we had this truth table. There were these 8 possible states, 3 genes, all right? So what you can, so what I want, so now what I want to do, so that was a situation where you actually altered one gene, right? So you reset the, like the state of the network, all right, and let it evolve from there. That's what we did in the previous case. Now let's say you want to alter the wiring of this network, all right? So because you don't like the steady state distribution that you have, right? You want to alter the wiring of this network. Now how do you alter the wiring? So let's keep these guys the same, these weighting of the predictors the same, but I can change these entries, right? I mean 0 to 1, I can change a 1 to a 0 or a 0 to a 1, right? How many possibilities are there? So see, like in each, so there are 5 columns here, right? We have 5 different predictors, right? 2 for gene number 1, 1 for gene number 2, 2 for gene number 3, right? So there are 5 different columns. How many possibilities do you have in each of these columns? How many possibilities? Okay, in this location, how many possibilities are there? If I'm going to change, how many possibilities are there? Yeah, but let's say okay, not for change, but how many possibilities are there of filling this one up? Let's say initially you're not given a value, right? How many possibilities? 2 possibilities, right? So there are 2 possibilities here also, right? So there are 8 of these. So for each column there are 2 to the power of 8 possibilities. That's 256 possibilities, all right? And then you have 5 different columns. So it's 256 times 5. I think it's like 1280 possibilities, okay? So what they're going to do in this case, all right, is you are going to try to make changes here, all right, in these entries, all right, and see if that gives you a better steady state distribution, right? So so you'll run through all the 1280 cases, right, that you possibly have, all right? Like, all the 1280 cases, like put in zeros and ones here. In each case, for each truth table, go and calculate the steady state distribution. All right, and then figure out, which one is giving you a steady state distribution close to what you want. That's the idea. So let me show you how that is done. And it's a brute force procedure, so it's not something really good. Okay, so this is the discussion, and let me get to that example. That's so in this particular example, right? Again, I mean, this with no perturbation, right? We had the state transition diagram. Then we put in a small perturbation probability 0.01, right? So that all states communicate. I can calculate the steady state distribution, and I found out, and I showed that to you in that vector, that for state 000 it was 0.07, and for this other 111, which is a bad state, it's pretty high, right? So in the long run, the network will be in this good state, which corresponds to quiescence only 7% of the time. It will be in proliferation 73% of the time. That's not good, right? We want to alter that. Say, for example, we want to make sure that each of these states has a steady state probability mass of only 0.4, right? So, and in that case, the other 6 states will be visited only 20% of the time in the steady state, right? So I want to accomplish that, right? So I actually run through all those different possibilities. See the 2 times 5 times 256 1280 possibilities, right? And then I'll go through this again next time because I'm doing it a little bit fast, okay? But I just want to show you the picture. So you run through, so there are 1280 simulations that you have done, okay? This is the steady state probability for state 000, steady state probability for state 111, right? And all these possibilities, each dot is basically a possibility. You're capturing that in here. And the optimal one, because you wanted both of them to be 0.4, so you run through this 1280 cases, and then this is the optimal one that you get, right? So that'll give you the optimal set of predictors that you want. I mean, I'm sorry, the optimal truth table that you want, right? And the only other thing that they do here is that, you don't want to go through all the 1280 cases, so maybe you can use some optimization problem to make your search narrower. Okay, so they use genetic algorithms, right? So this is another paper. And again, on this, just look at this stuff, all right? If there are any questions, ask me next time, right? Yeah, let's stop here for today. Right?